This study follows on from Lucy's study in some ways, in that we were looking at the bacteria species that were associated with the urothelial cells from patients with refractory Detrusor overactivity. And so recently, as Lucy's just mentioned, infection is quite common in the urinary tract of patients with refractory DO. And what we've found in our group is that when the patients with refractory DO have an exacerbation of their symptoms, then they're more likely to have an infection happening and have bacteria present. The problem is that we know that some of these uropathogens can form intracellular bacterial communities, particularly the uropathogenic E. coli, and that's difficult to demonstrate. It's time-consuming to do the confocal microscopy, and so this image here is a confocal microscopy. The red is the weak germ agglutinins showing the outside of the urothelial cell, and the small bright blue dots are the intracellular E. coli. So the aim of this study was to try and examine the way the bacteria are associated with the urothelial cells using a simple light microscopy technique, and then to compare the bacteria that we saw associated with the urothelial cells with what the lab reported on routine microbiology. So we collected midstream urine samples from 39 women with refractory Detrusor overactivity. Half of the urine sample went to the microbiology lab for routine clinical testing, and the other half went to the research laboratory. And we concentrated the urothelial cells onto a microscope slide, stained them with right stain, and then examined them by light microscopy. The first finding was that the routine micro lab detected bacteria in about 76% of the patients, where when we examined the urothelial cells by light microscopy, we could see bacteria in almost all. 97% of the patients had bacteria associated with the urothelial cells. When the routine micro results came back, they told us which was the dominant organism, and the most common dominant organisms were the E. coli, Enterococcus and Streptococcus, with 38% being E. coli, 17% being Enterococcus, and about 26% being the Streptococcus. But we weren't really interested in the exact bacterial strain that the lab reported, but rather how those bacteria were associated with the urothelial cells themselves. So to characterise that, we selected 100 urothelial cells on the right stain and then described the association of the bacteria with the urothelium. So firstly, we had bacteria that appeared adjacent to the urothelial cells, and then low or high density. So here you can see the bacteria ringing the outside of the urothelial cells, so appearing adjacent and high density because the bacteria were covering more than 50% of the rim of the urothelial cell. And then appears intracellular, which is where we had either scattered bacteria inside the urothelial cell, or we had a urothelial cell that had a lot of bacteria associated with, looking as though it could be inside of the cell. So remembering this is light microscopy, so it appears intracellular. We couldn't prove that it was definitely intracellular because we hadn't done confocal at this time. So these are the results that we saw, and as you would expect with the E. coli bacteria, that's a known uropathogen to form intracellular communities, it had a lot of bacteria that appeared intracellular, so more than 60% of the cells that we examined from the patients with a E. coli predominant infection appeared intracellular. But what we also found was that about 25% of the patients from Enterococcus or Streptococcus infections also had bacteria that appeared intracellular. So if we summarise it in this table, with the E. coli, about 88% of the cells that we examined were associated with bacteria, and about 66% of those were appearing intracellular. A smaller number of cells from the Enterococcus and the Streptococcus were associated with bacteria, but we still had a reasonable number that appeared to be intracellular. So what we found is that the simple right staining with light microscopy was able to detect more bacteria than in the urine samples than the routine microbiology would determine, and that we were able to show bacteria that appeared intracellular with all of the three species that we looked at, with the Enterococcus, with the Streptococcus, and then with the E. coli, which we expected to see. Thank you.
So any question from the audience? So in your finding, if the patient has intercellular bacteria is a large amount. How do you treat this patient?
Yeah, it is. It does make it a bit more complicated to treat them. So looking for antibiotics that will be able to permeate the cell. But then also we believe that these bacteria are fluxing in and out of the urothelial cells. So what we've found is that when the patients are having an acute exacerbation, that they're more likely to have the bacteria. So sort of a long-term type treatments, like what James Malone-Lees described in the past, that long-term treatment to try and remove the bacteria when they're out of the cells.
How long?
Yeah, that's the question, isn't it?
And need any combination therapy or two kinds of antibiotic or something?
That's what we would be recommending, is looking at a combination of different types of antibiotics so that you could be, a lot of the bacteria, as Lucy was saying, a lot of the bacteria have antibiotic resistance, and so making sure that you chose the right one to get all of the different strains.
Thank you.
Yeah, Zeran from Dallas. I was intrigued by your slide where you showed a difference in bacterial recognition between you and your microscope and your lab. Have you sent to different labs to see if it's just bad luck, or is this a general issue that we have to be concerned about? Because I think clinically we see that. People get urine culture, it comes back negative, the symptoms are there, they ultimately respond to antibiotics, so our level of detection is not sufficient. That's a really clinical issue. It's a great slide. I was just wondering if you'd further study that and send it to other labs to see if you find the same thing, or just those two places.
We haven't routinely sent samples to other labs. We have, when we've collected samples from the university where I am rather than where the rest of the group is, that's gone to another lab and we've seen the same findings, but we don't routinely send them to numerous places. But when we've discussed it with the micro lab, they think it's more to do with the fact that we've concentrated what we're looking at. Because we're spinning down the urothelial cells, they're just taking their 10 microlitres off the top of the urine sample, where we're concentrating it down so we can see just the very small numbers that are there, where they're only interested in a large number of bacteria.
The other question I had, you seem to have an easy breakdown between intrabacterial and non-intrabacterial, but do you have a blend when you look at these cells and can you really easily classify patients being in one group or the other, or it's more like 20% have this and 30% have that?
Some of the, sometimes when you look at the slides, they'll have adjacent bacteria and intracellular bacteria. Sometimes you'll have bacilli and cocci all in the same slide. So yeah, it's more complicated than a simple presentation. And we've done low density and high density, but even that took us quite a while to figure out how we're going to define low and high density, because we couldn't say for the intracellular ones, okay, so it covers 10% of the surface or 20% of the surface or 50% of the surface, or so sort of finding some way to grade them took quite a long time.
Yeah, that was my next question. So is this reproducible? If someone else other than you looks at it, they could come up with the same scoring, so to speak, or have you not tested that?
We've tried that. So we've had about 3 or 4 people in the lab who've looked at similar samples to see if we come up with similar scoring, and we do tend to come up with quite similar things. But you have to be quite specific about what you're going to say is low density, high density.
Great. Thank you again.
Thank you.
Chris Payne from California. Just a quick clarification. These are Detrusor overactivity patients who clinically are not infected. Is that right? I mean, do they have pyuria? I guess that's really my question.
Yeah. So these are patients with refractory DO. Some of them, I have got the pyuria data, but it's not in my head at the moment. Some of them will have pyuria because, like when the microlab reports back on the bacteria, some of them will have had pyuria as well. But we were more interested in the bacteria than the pyuria.
Okay.
So
last question. Okay.
Sure, John Lavelle from Palo Alto. Just a simple question first. Is these samples for the cells are all taken from the spun urines of the MSU, right?
Yes.
So in relation to the findings, you're suggesting that these findings are maybe what's going on within the bladder itself, but yet you have no evidence that this is actually happening in the bladder wall. So potentially these bacteria could be found around the cells, but they may have collected, say, overnight, if this happened to be the first void in the morning or something similar.
So we don't, our samples aren't usually first voids because
Yeah.
the patients are coming into the clinic, so they've usually voided before they've come into the clinic. And so they void in the clinic and it goes straight to the laboratory, so within about 5 to 10 minutes it's spun down. And the other thing that we've been really careful of is that we're spinning at quite low speeds, because if you spin at a really high speed, any bacteria that are just floating around not associated with the cells will also be spun down. So we're only interested in the bacteria that come down because they're attached to the cells. So we're spinning at quite low speed that will bring down the urothelial cells, but not necessarily the little tiny bacteria.
Yeah, but these are the free-floating cells found
Yes,
in the
they're the
urine.
free-floating
Yeah.
cells,
So it
yes.
may or may not relate to what's actually going on in the bladder wall
Yeah, we
itself.
haven't done it on biopsies or anything yet.
Okay, that's your question. Okay.
Dr. Gutman from Turkey. Are there any difference between two groups of bacteria that you determine with normal routine way or microscopically according to the antibiotic resistance?
We haven't looked at the antibiotic resistance in the bacteria that we've isolated with the urothelial cells. So that's something that we could do moving forward, would be to spin down the cells and then plate those out instead of what the lab would normally plate out, but we haven't done that yet.